All right, so the regulation occurs in that if there is lactose, so there's food around, and the E. coli has to make use of it. And the E. coli now has to express these three genes, these three open reading frames, that gives rise to a single message RNA, all right, that gives rise to three different types of protein, which we said earlier on has unique roles in terms of uptaking, modifying it, and breaking it down. So if lactose is present, these genes are expressed. If lactose is not present, then these genes aren't expressed. And nature is very efficient because if it doesn't require it, it won't make it. Right? It wants to be, has a maximum efficiency. So it's only when lactose is around that these genes are expressed. And if there's low lactose, then why make it? Okay? Same thing for the trip operon. Genes will only express if there's a need to make tryptophan. If there isn't, the genes aren't expressed. Right? This type of on and off is called gene regulation. Okay? Regulation, to regulate, meaning to turn on or turn off the gene. All right, now, here's where the fun begins. Okay, we understand about the lactose, the lac operons, and we said, okay, we have an open reading frame, and we know the sequence of those because we can look it up. We can know exactly the ATG sites that are here. We know that each of these have unique ATG sites, okay? And we know exactly the sequence of the promoter and the operator. Okay, now let's have a little game. Let's say now, I want you to make a bacteria, a modified bacteria in our case, in such that if you add lactose in the media, the E. coli fluoresces green. All right, so we're mixing two things that naturally does not happen. Right? So naturally this doesn't happen. You don't have an E. coli that fluoresces green. But let's say you want an indicator that says, okay, if there's lactose around, then I want the bacteria to turn green. How do we do this? All right? Now, you could say, well, what's the practical reason for this besides, besides just doing it, right? I mean, one could say, well, if lactose is something you don't want, it's an indicator of it. So perhaps lactose-free milk, if you really want to check if it's truly lactose-free, and if you want to make sure you don't get a stomachache, you might
just dump some E. coli in first and then drink it.
We'll put a little, actually the other way, put a little milk into the E. coli and see what it flows.
So you don't really, you don't want it to break it down, you just want it to fluoresce if it's there?
So yes.
So just cut out that part and put in the...
Yes. So here's our design. We want to know if in the presence of lactose, who cares about making all that? All right. We don't care. The bug will starve to death, but before it starves to death, it will give off green. Right? We're very insensitive people. We don't care about the E. coli. Right? But what we do want to know is that in the presence of lactose, can we make the E. coli green? And how do we do this? Okay. Yes.
Add another reading frame after the site.
Okay, yes. So now we're talking about not only the open reading frame modification, but we're talking about promoters as well. This is a promoter operator, and this is a regulator. These are what's important to turn on what's down here. It actually doesn't matter what's down here. Okay, it just happens naturally. It belongs to the three open reading frames. But you could actually put almost anything in there, and it will express it. If it's not toxic to the E. coli, it's able to make it in full. So if we know the exact sequences of this whole operon, then what we can do is construct it in terms of having that whole green fluorescent protein and pop it right in there, have it in frame. All right, so let me show you just to give you a grasp, make sure we understand how the lac operon operates, because what we're going to do is start designing this bits and pieces. We got the green fluorescent protein, now we're going to play around with this operator site. So,
Oh,
The
yeah.
E. coli lac operon is an example of an inducible set of genes. These genes are responsible for the breakdown of lactose into sugars used for cellular metabolism. This inducible system also involves bacterial DNA, a repressor, mRNA, and the sugar molecule lactose. This animation will only focus on 2 of the 3 proteins encoded by the lac operon: beta-galactosidase and permease. Gene expression can be induced or turned on when a specific inducer molecule appears in a cell. For inducible systems, a repressor molecule prevents gene expression by binding to the upstream controlling region. Lactose is the lac operon inducer molecule. After first appearing in the cellular environment, lactose passively enters the E. coli cell and binds to the repressor molecule. This binding releases the repressor from the controlling region. At this point, RNA polymerase can begin transcription of the operon. Here we show 2 of the 3 lac operon genes being transcribed into mRNA. Ribosomes then bind to the mRNA and the 2 proteins are translated. The first protein is beta-galactosidase, which breaks down lactose into 2 simple sugars. The second protein is permease, a membrane-bound protein.
so imagine green fluorescent protein in its place.
Permease functions to provide the direct route for the lactose outside the cell to be imported into the cell. This import occurs at a much greater rate than the passive transfer we first observed. Because translation continues inside the cell, other permease proteins become embedded in the membrane. This further increases the rate at which lactose enters the cell. Beta-galactosidase breaks the cellular lactose into the simple sugars glucose and galactose. Once its concentration is greatly reduced, the lactose bound to the repressor are released. At this point, the repressor again binds to the controlling region and gene expression is halted. For all inducible systems like the lac operon, it is the interaction of the repressor and inducer molecules that mediate gene expression.
Okay, so for this class purposes, really not focused on the lac operon, but it's focused on using the lac operon promoter and a regulatory site to turn on and off of a gene that we want. And the gene that we're focusing on is green fluorescent protein, and because when it's expressed, you could actually see it. Okay, now, any questions up to this point? Okay, we're okay with this. All right, now, I'm going to go back to the plasmid and tell you some interesting points about this in addition. Okay, so we said, here's the ribosomal binding site. And the ribosomal binding site, we said it's important because this site is important for recognition of the ribosome, for translation, right? It's not really important for transcription per se, but for the subsequent translation, this sequence, at least the RNA version of this is really important. We also said that the promoter here is important, and this promoter is a T7 promoter. And as I said before, the T7 promoter here is actually from a viral promoter, and in this whole system, this plasmid, it is an expression plasmid, meaning that if you have this gene, whatever gene is in here, and this also has a modification of a lac type of operon towards it. In fact, the way this whole system works is that if you put a gene in here into this pet plasmid and transform it into E. coli, right, you can actually put let me Just put this up here. In the E. coli, this particular E. coli strain that we have, so I'm going to draw the genomic DNA as this. So this is genomic DNA. And the chromosome that we've added, recombinant chromosome, is here. Right. In that recombinant chromosome, we have the targeted clone open reading frame. And in that genomic DNA, this particular strain of E. coli has been designed to, engineered to be sensitive to a lactose analog, right? So this inside here has a very similar feature of an I protein like the lac alpha-1 repressor, and it produces the repressor, and it's sensitive to also lactose as well. But an analog of this is called IPTG, which stands for isopropylgalactoside. And it's an analog of lactose, meaning that it looks like lactose. It will bind to the same protein like lactose, but it will not cleave like lactose. So just to give you a reminder of how this may look, here's lactose again. Draw this out. So it's 2 sugar molecules together. This is bound. And so here's galactose. Okay, so here's galactose and glucose together. Isopropylgalactoside is very similar. It has a sugar of galactose that is now bound to a sulfur isopropyl. Okay, so that's IPTG, right? And chemically, this repressor here cannot distinguish between these two. It will also bind. Okay, so if you have IPTG around, this will bind, but it will not cleave. So that means that it will last longer. So in this expression system, normally what we do is we put the recombinant protein in there, and when the cell is growing, we add IPTG. And when we add IPTG, the protein expresses. And the reason it expresses is because once this is blocked, right, the RNA polymerase can now bind onto the promoter and goes through transcription.